Hi guys, and welcome to our module on connective tissue 2. As we mentioned in the previous module, the connective tissues are one of the four major tissues found in the body. The rest would include the epithelial tissues, the muscle tissue, and the nervous tissue. The uniqueness of the connective tissue is in the fact that it consists of both organic and inorganic elements that are largely derived from embryonic mesenchyme and tends to underline the epithelium and subserve metabolic and structural functions. We talked about some of the features of connective tissue, and we also classified connective tissue. Just to remind us about some of the characteristics of connective tissue, um, we need to remember that it is made up largely of extracellular matrix with few cells embedded within it. So we can say the components of connective tissue include cells and extracellular matrix, but largely the extracellular matrix. The cells are divided into wandering cells and fixed cells, whereas the extracellular matrix includes ground substance and extracellular fibers. We talked about this in at a larger extent in a previous model, that's connective tissue 1. And in terms of the division or the types of connective tissue we have, we mentioned that we could divide it into the connective tissue proper and the specialized connective tissue. In the previous model, we talked about the connective tissue proper. For this particular model, we'll be focusing on the second aspect of the connective tissue, which is the specialized connective tissue. So the connective tissue specialized or the specialized connective tissue is divided into three major types. This includes cartilages, bones, and blood. And we'll talk about each of these different forms of specialized connective tissues in this particular model. Starting off with blood. So blood is a liquid or fluid specialized connective tissue. It is found within the cardiovascular system and it's a circulating medium within the cardiovascular system. It's pumped by the heart and circulated around the body by vessels. And its particular function is in the delivery of different substances or molecules within the entire body. So the functions of blood include functions of distribution, or self-sealing, disease and infection fighting, and acid-base balance. So, for example, in terms of distribution, the blood is involved in distribution of nutrients to the various organs of the body, waste to sites of excretion, hormones to various target sites, gases like oxygen and CO2 to the sites where they are needed or where they are excreted, and then drugs, depending on what particular drugs we are using. um, then the function of self-sealing is just a fancy way of saying the blood is also involved in the process of hemostasis or clotting. We'll talk about that extensively in a separate module. Then for disease and infection fighting, blood contains unique cells we know as immune cells. So cells like white blood cells are important um, for immunity. Not just even cells as well. Proteins like antibodies are found within blood are also important to provide protection against germs. Um, then we have the acid-base balance. Acid-base balance, blood is involved in maintaining the acid-base level, as it ensures the medium for optimal performance of enzymes, organelles, and cells by maintaining the amount or the range of pH present within it. It does this by the presence of buffers. So there are protein buffers and bicarbonate buffers found in blood that helps in regulating acid-base balance. Let's talk about the components of the blood. And just like how we mentioned the components of the entire connective tissue, we use this as a template in talking about the components of the different forms of specialized connective tissue. So remember that the connective tissue has its components being divided into cells and extracellular matrix. In the case of blood, this is not different. So because blood is a connective tissue, it will also be composed of cells and an extracellular matrix. The extracellular matrix of blood is largely fluid, and we know this as plasma. So plasma is the name of the extracellular matrix. And this extracellular matrix can further be divided into ground substance and extracellular fibers. In the case of blood, the ground substance of blood is just known as serum, and the extracellular fibers is largely known as fibrin. So we can further divide the plasma into fibrin and serum. And we'll talk about each of the different components of blood in the next slide. So to further break down the components of blood, the cellular component of blood includes three major forms of cells. We have the red blood cells, the white blood cells, and the platelets. The extracellular matrix of blood, I talked about the division already. It's divided into serum and fibrin. Let's start with the extracellular matrix of blood. Plasma is the fluid by which the cells are usually suspended in. It's the medium in which the blood cells are transported around the body, in which circulation tends to occur. Is what makes blood to be largely fluid. Plasma is composed of three major components. It's composed of largely water. 92% of plasma is water. Um, of plasma protein as well. Proteins are found within plasma in large amounts. And then solutes. So solutes would range from anything from ions to carbohydrate molecules to fatty molecules. All these are solutes found within blood. So about 92% is water, about 7% is protein, and then about 1% is solid. This is a picture of how we divide them. So you can see plasma being present in large quantities in terms of proportion of the entire blood. Then focusing on the cells, we have each of the cells also making different proportions of the entire blood. So red blood cells takes about 99.8% of the entire cell. White blood cells about 0.1%, and platelet takes about 0.1%. So red blood cells are also known as erythrocytes. They are the most abundant blood cells in the entire blood. And their major function is in the ability to carry oxygen as well as carbon dioxide, but largely oxygen. And they do this by virtue of the fact that they contain an iron compound called hemoglobin within them. This iron compound is usually what makes red blood cells reddish in their color. White blood cells are cells that are also called leukocytes. They are immune. They are the immune cells of the blood. They are involved in the protection of the body against germs. We tend to divide white blood cells into two forms. We have the granulocytes that contains large amount of cytoplasmic granules within them, and we have the lymphocytes, which are cells that tend to migrate in and out of the blood into special lymphoid tissues. So granulocytes are called granulocytes because of those visible cytoplasmic granules. We also have some cells that are not granulocytes, that are what we call agranulocytes. They are not really classified as lymphocytes, neither are they granulocytes, but they are in between. So they call them agranulocytes. So they don't contain cytoplasmic granules. And so these examples of the cells would include the monocytes. So this is just a broad division of the entire white blood cell. So we see granulocytes. Monocytes are the cells that we talked about that are not necessarily granulocytes. They don't they fall into the category of lymphocytes, and they're just by themselves. And then we have the lymphocytes. So to divide them further, the granulocytes divided into neutrophils, eosinophils, and basophils, and then the lymphocytes will be divided into T cells and B cells. So these are just blood film images of the different forms of white blood cells we have. So we just use it to talk about some of them and the peculiarities between them. So starting with the neutrophils. Neutrophils is the image shown directly here. These are polymononuclear cells, and what I mean by that is that they are cells that contain large amount or large number of lobulated nucleus. So if you look at this, the nucleus found within the neutrophils is multiple. You could have about 3 to 5 nucleus present there. If you count this would be about 3 to 5 nucleus in this, or even more than that in this particular image. So once you see a cell that has multiple lobes of nuclei, you should know that that is the neutrophils. Neutrophils makes up about 70% of the entire white blood cell, so it's about 2/3 of the circulating white blood cell. It contains cytoplasmic granules, which is the reason why we classify it as a granulocyte. And this cytoplasmic granules has lysosomal enzymes present within it. The Granules are usually pale in appearance, and we say that they are neutrophils because of the fact that the granules usually tend to just have a neutral spin and not necessarily either reddish or bluish stain, which is in comparison with the other two granulocytes. The major function of the neutrophils is its ability to fight against bacteria, and how it does this is through its phagocytotic action. It has the ability to undergo phagocytosis, and the particular target germ that is involved in protecting us against would be bacteria. Then moving on to the second image, this image shows depicts for us an eosinophil. Eosinophil is um, compared to a neutrophil, tends to be bilobed. So we have the nucleus being bilobed. We have one and two lobes of the nucleus. It makes up about 2 to 4% of the circulating white blood cell. It tends to have its own fair share of cytoplasmic granules, but the cytoplasmic granules of the eosinophils usually stains with eosin, so it has a reddish stain. And the function of eosinophils tend to be in its ability to fight against parasitic infection. So the way in which neutrophils fight against bacterial infection, eosinophils is going to be involved in fighting against parasitic infection. Aside that, eosinophils have also been noticed to work more in the scenarios of allergies. So allergic reactions, we see a lot of eosinophils being involved in it. The third picture here shows the white blood cell that is regarded as a basophil. Basophil are cells that are also bilobed, so they are bilobed in terms of the amount of lobes of their nucleus. They have two. They make up about less than 1% of the circulating white blood cell. Their granules tend to stain with basic dye, and that's the reason why they appear bluish compared to the eosinophils. So neutrophils appear neutral, um, eosinophils appear reddish or pinkish, and basophils appear bluish. And they contain the cytoplasmic granules contain large amount of histamine, and this will play a role in one of their functions. Usually, basophils are involved in allergic reactions largely compared to eosinophils. And one of the ways we know this is the fact that the cytoplasmic granules contains histamine. Histamine tends to be discharged at inflammatory sites during this anaphylactic or allergic reactions. And basophils tend to migrate out of the blood cells into tissues. When we have a basophil found in tissues, we usually refer to it as a mast cell. So mast cells are just basophils found in tissue. The last image in this particular slide is that of a monocyte. Monocytes are not granulocytes. Okay, if you notice, they don't have granules present within them. They make up about 2% to 8% of the entire white blood cell. They have a unique nucleus. They don't have a multi-lobed nucleus or bilobed nucleus as we see in neutrophils and the other granulocytes. But the nucleus tends to be kidney shaped. You can look at this nucleus; it has a kidney shaped appearance. The function of the monocyte is in its ability to cross over um, from the circulation into the tissues to become a macrophage. And macrophages are there in immune cells in tissues that have phagocytic activity. They are involved in phagocytosis just like the neutrophils, but they do this within tissues. So we have different monocytes becoming different tissues, have different macrophages in different tissues, and they have different names there. So for example, the macrophage in the lungs would be referred to as the alveolar macrophage. Macrophage in the kidney is referred to as the mesangial cells. Macrophage in the brain is known as microglia cells. Macrophage in the skin is known as dendritic cells. In the spleen, we know them as histiocytes. In the lymph node, we know them as bile cells. So we have different names for the different macrophages in different sites. The last image here is that of the lymphocytes. The lymphocytes are not granulocytes and neither are they monocytes. And we could see how they distinguish themselves to the other cells. They make about 20 to 30% of circulating white blood cells. So they are next to neutrophils in terms of their proportion. And they are relatively small cells. They are slightly larger than red blood cells. And what is unique to note about them in terms of how we distinguish them from the rest of the cells is that they have a very large nucleus. You can see how round and large the nucleus almost takes about 90% of the entire cytoplasm. And this is in contrast to the monocyte. If you are trying to distinguish between a monocyte and a lymphocyte, monocyte has its neutrophil making about a third of the entire cytoplasm, not the entire thing, but two-thirds. And the nucleus of the monocyte tends to be um, kidney shaped. For lymphocyte, the nucleus is round and takes up about 90% to 98% of the entire cytoplasmic space. Lymphocytes are divided into two forms. We have the T and the B lymphocytes. Their functions: for the T lymphocytes, it is largely involved in fighting viral infection, so viruses. It's also largely involved in fighting cancer cells or tumor cells. For the B lymphocyte is involved in fighting bacteria, and it doesn't do this directly. It does this by the production of special protein we know as antibodies. So antibodies are produced by B lymphocytes, and the antibodies are involved in the fight against bacterial infection. The last blood cell would be the platelets. Platelets are also known as thrombocytes, and platelets perform the function of the self-sealing I mentioned, which is the ability to undergo hemostasis of blood clots. Platelets are involved in blood clotting. They help in reducing the loss of blood at a particular location where injury has occurred, and they do this through the process of hemostasis. So bones are specialized connective tissues that are involved in forming the skeletal system or skeletal structure. They are connective tissues with the extracellular component being mineralized. So if we are comparing them in terms of blood is a specialized connective tissue that has its extracellular component being unique in the sense that it is fluid. For bone, the extracellular components of bones are mineralized, and that's what makes it unique. One important property of bone is the fact that it is rigid and it has marked or strength and some degree of elasticity. We'll talk about the different components of bones and its function, as well as some other aspect of bones like formation and its classification. So the function of bones. Bones has multiple functions. First is hemopoiesis. Bones have particular structures within its space known as a marrow. So bone marrow is a site where blood cells are formed. So you can see how blood and bones are connected. The marrow within the bone serves as the site of production of those blood cells we mentioned when talking about blood. So white blood cells, red blood cells, and the platelets are formed within the bone marrow. Aside that, the bones are involved in supporting body weights because of the fact that they're involved in forming the skeletal framework. They're involved in protecting soft tissue. So bones like the cranium is involved in protecting the brain. Bones like the ribs is involved in protecting the lungs and the heart. Bones like the pelvic region is involved in protecting structures like the genital organs. Bones are also involved in movement at different joints. Two bones join together to form a joint. We'll talk about joints in a separate module, but at joints we have movement occurring. Bones are involved in storing of calcium and phosphate. Remember that the extracellular component of a bone is largely mineralized, and the minerals we see within this extracellular component contains large stores of calcium and phosphate. And sometimes when we do need calcium and phosphate within the blood, we could have bones releasing the calcium and releasing phosphate into the blood to maintain calcium and phosphate homeostasis. So what are the components of bones? Just like other connective tissues, bones are divided in its components into cells and extracellular matrix. The extracellular matrix is further divided into the ground substance and extracellular fibers. So in terms of talking about bone as a structure, bones have cells and have extracellular matrix. The extracellular matrix of the bone is largely mineralized. It has its own set of ground substance and as well as um, extracellular fibers. The extracellular fibers we see within bones is made up largely of collagen, and this is what provides it its strength and tenacity. So, talking about the different components and breaking it down further, the cells found within bones are divided into five major types. We have the osteoblast, the osteocyte, the osteoclast, the bone lining cells, and the osteoprogenitor cells. The extracellular matrix would also be divided into different forms. We have the ground substance having organic compounds and inorganic compounds. The inorganic compounds is what we talk about when talking about the mineralized form. And then we have collagen fibers being the major extracellular fibers in a bone. Let's focus on the bones or the cells of the bones. So we'll talk about the different types of cells: the osteoblast, the osteocyte, the osteoclast, and the bone lining cells, and the osteoprogenitor cells. So let's start with the osteoblast. Osteoblasts are basophilic, cuboidal, mononuclear cells that are found within the bone matrix. So they're found within the bone matrix surface. They are non-dividing cells and they tend to perform the activity of what we call osteoblastic activity. Osteoblastic activity is just what we call the bone forming activity. So they are involved in forming the uh, production of the extracellular matrix around which the bone is formed. So they are involved in building up this ground substance and building up the collagen and also adding extras to the mineralization. So they do the bone forming activity of a bone, and that just involves the mineralization of the bone as it entails. They contain particular organelles that are involved in its function. They have large amount of rough endoplasmic reticulum, Golgi apparatus, and dense particles that is rich in calcium. And like I said, they're involved in the synthesis, and deposition, and mineralization of the bone matrix. And because of this type of organelles they have, the rough endoplasmic reticulum, the Golgi apparatus, and then the calcium particles they have, that's what makes them be able to perform those functions I mentioned. So newly secreted organic matrix tends to be formed by the osteoblast. And we usually call this newly secreted organic matrix osteoid. And then this is now further mineralized by the osteoblast to give us the final mature bone matrix. When we have deficiencies of vitamin D or calcium, it usually results in poor mineralization. And we see this in particular conditions we know as osteomalacia. This could also arise when there's a problem with osteoblastic activity. So when osteoblasts are not doing their function well, you could have osteomalacia occurring as a result. So once osteoblasts are formed, and one osteoblast starts to form extracellular matrix that becomes mineralized, it becomes or slowly becomes the second bone cell, which we know as osteocytes. Osteocytes are just mature osteoblasts. So what do I mean by that? So imagine an osteoblast forming the osteoid, which is the immature matrix around it, and then now mineralizing this matrix. Once the mineralized matrix is formed, this matrix is surround the osteoblast. The point in which we have the mineralized matrix surrounding the osteoblast, we stop calling this the osteoblast and we start to call it the osteocytes. So osteocytes are osteoblast, mature osteoblast surrounded by mature matrix. So we usually see that they are the major cell types we see in mature bones, so bones that have been already formed, mature matrix. Cells found there will be largely osteocytes. And the space in which they tend to lie in is known as the lacunae. So lacunae is the space in which the osteocyte finds itself after the mature matrix surrounds it. The function of the osteocyte is in the performance of osteolytic activities. All right, and how it does this is by maintaining the extracellular matrix that is found around it. So osteoblasts will build the matrix, osteocytes would maintain the matrix. The difference between osteoblast and osteocyte in terms of their cytoplasmic processes is in its is in the reduction of rough endoplasmic reticulum and Golgi apparatus. Some of those things we mentioned, they are reduced in osteocytes compared to osteoblast. The osteocyte also has cytoplasmic processes, what we call philopodias, um, that are involved in. So if you look at a picture like this, you can see some of these extracellular processes. These are philopodias, the extensions or cytoplasmic extensions of the osteocyte. They extend into the mineralized matrix channel and they form what we call canaliculi. They are the parts that are involved in direct maintenance of the matrix around the osteocyte. Osteocytes also permit the release of calcium into tissues. Remember, I said that one of the function of bone is in the maintenance of calcium, and if we have bone, the blood being reduced, if calcium being released within blood, bones can release this calcium into blood to maintain its level. So one of the ways in which this is done is through the release of calcium from the osteocyte or through the osteocyte into blood. Um, Parathyroid hormone plays play a role in this, and the osteocyte contains high amount of receptors for parathyroid hormone. So parathyroid hormone, once they bind to the receptors of the osteocyte, they will promote osteolytic activities and help in releasing calcium into the blood. The third bone cell would be the osteoclast. Osteoclasts are multinucleated cells. So you look at this, they have multiple nucleus found within them. They are multinucleated bone cells. They are located in small cavitations known as house-shaped lacunae. So they are also found in lacunae, but this is smaller compared to that of the osteocytes. They are also involved in performing osteolytic activity. But the type of osteolytic activity that we see in the osteoclast is a bit um, extensive compared to that of the osteocyte. So, osteoblast is involved in building the extracellular matrix. Osteocytes are involved in maintaining the extracellular matrix, but osteoclasts are involved in breaking down the extracellular matrix. They are involved in matrix breakdown. So, um, usually these osteoclasts are formed by the fusion of monocytes, and they tend to um, be rich in lysosomes. That's why they are also involved in breakdown. So they are rich in lysozymes. Lysozymes are usually going to be involved in breakdown of different structures. They also have numerous cytoplasmic projections, also which are known as Ruffeld bodies. So these are Ruffeld bodies found in the osteoclasts. So it's similar to what we see in the philopodia for the osteocytes. So you can see that both of these cells are involved in osteolytic activities, have some cytoplasmic processes. For osteocytes, it's the philopodia. For osteoclasts, it's the Ruffle bodies. What we need to remember about the osteoclast is the fact that it's involved in an extensive matrix breaking or matrix resolving function, in contrast to the osteocytes. And this is also promoted by parathyroid hormone. So parathyroid hormone will work on both the osteoclast and the osteocytes. Osteocytes, it does that to maintain the extracellular matrix. For osteoclast, it does that to resolve or break down the extracellular matrix. The osteoid progenitor cells are practically the stem cells of the bone. They are the cells that are pluripotent in nature, and they are involved in forming every other stem cell. So you could see in this image how the osteoid progenitor cells are involved in forming osteoblast and osteocyte and even osteoclast. They are largely just the pluripotent cells, stem cells found there. Finally, we have the bone lining cells. These are flattened epithelial-like cells that are found on the resting surface of bones. They are involved in three major things. They are involved in regulation of differentiation of osteogenic progenitor cells. They are involved in access of the osteoclast to bone surface, and they are also involved in the mineralization or hemostasis or mineral hemostasis. They maintain the amount of minerals that gets into the bone as a result. Let's talk about the second component of the bones, which is the extracellular matrix. The extracellular matrix of bone, like I said, is divided into two parts: the ground substance and the extracellular fibers. The ground substance is composed of organic and inorganic compounds. The organic compounds in ground substance include things like glycoprotein, like gelatin. The inorganic substances of the extracellular fiber matrix contains salt and mineral-like structures, which could be calcium phosphates, calcium carbonates, calcium fluoride, magnesium phosphate, and then sodium chloride. You notice a lot of calcium and phosphate are found within the inorganic compound, and that's the reason why we say they tend to store large amount of calcium and phosphate within them. The extracellular fibers found within the extracellular matrix of bone is predominantly formed by collagen. Collagen maintains the tensile strength and the integrity of the bone. Let's classify bones. So we can classify bones according to the fiber arrangement we see. And based on the fiber arrangement, we classify bones into a woven bone or a laminar bone. So if you look at the picture shows us the different forms of bone, and this is based on fiber arrangement. Remember that when we classified the dense connective tissue. We also classified it based on fiber arrangements. Remember that dense connective tissue could either be irregular or irregular, and this is based on how well arranged the fibers are. In terms of bone, woven bone and laminar bone could be also be classified that way. So laminar bone has well arranged fibers. Woven bone have usually irregularly arranged fibers. So woven bone, immature bone, they are the first bone that is initially produced. They have randomly organized fibrous elements. So the collagen found within the woven bone is randomly arranged. You can see how woven it is or woolly it is. And this include bones that are found in these are found in include bones found in tooth sockets, in sutures, and the bones of the embryo. And that's the reason why we say they're usually immature bones. Lamellar bone are mature bones. They tend to have the fibrous elements being uniformly and regularly organized, as shown in the image B. They are involved in forming bones that we see in the adult structure or in a child that has just been born. So bones that are found in every other aspect of the body, aside those areas I mentioned, like the suture line, the suture site, the tooth sockets, and in the embryo. They are formed from the remodeling of the woven bone. So woven bone would eventually give rise to lamellar bone. This is due to remodeling occurring. So bones are usually organized into three layers. We have the entire a single bone having three major layers macroscopically. So these layers includes the outer layer, or we call the periosteum, the middle layer known as the bone tissue proper, and then the inner layer known as the endosteum. We talk about each of these layers of the bone little by little as we move on along the slide. So you can see a picture of those layers. You can see the periosteum here. We can see the endosteum present within the image shown here, and then in between that those two layers we have the middle layer or the bone tissue proper. So the radium is the outer part of the bone, covers the entire bone surface. It's composed of two major sublayers, an outer fibrous layer and an inner cellular layer. And it's essential for bone growth in diameter and also for fracture repair. So note that the periosteum does not cover the articular surfaces of a bone, and it tends to be the site of tendon attachment. So the articular surface of the bone is usually the site of the bone that is in contact with another aspect of the bone involved in the formation of joints. So wherever we have joints, we have the articular surface of a bone, and that part is not lined by periosteum. These fibrous part or the fibrous part of the periosteum tends to be highly vascular, whereas the inner part or inner cellular layer is usually composed of osteogenic cells. And the um, the periosteum tends to contain the large amount of collagen fibers that connects it to the bone tissue proper. So that those collagen fibers that connects the periosteum to the bone tissue proper is usually known as the Sharpey's fiber. So Sharpey's fiber connects the periosteum to the underlying middle layer. So let's talk about the bone tissue proper. Bone tissue proper is the middle layer of bone in terms of organization. It forms the bulkiest part of the bone structure. We have it also being divided into two parts. We have the compact part, which is the outer portion of the bone tissue proper, and we have the spongy part, which is the innermost part of the bone tissue proper. So the spongy part of the bone is also known as the cancellous bone. It is usually described as a network or trabecular network of bones that are intervening and forming spaces in between them. Usually, this spongy portion of the bone usually is filled with marrow. It's filled with the marrow that is involved in hematopoiesis, or what we call hematopoietic marrow tissue, and that's by virtue of the fact that the spongy part has spaces between them. So it's not a solid structure in it. So just think about it as like a sponge. Sponge has those different networks with spaces intertwined between them, and as a result, that's how the spongy bone tends to be. Spongy bone or spongy or cancellous bone is seen in flat bones of the skull. It's seen in bones like the sternum, like the ribs, like the epiphysis and metaphysis of long bones, and the alveolar bones of the around the tooth. The second is the compact or the cortical bone. So compact bone is also called cortical bone. is found on the outermost layer. So if we were to divide it, if we were to organize it again, we have the periosteum. Periosteum being divided into fibrous part and inner part. Then after periosteum is the bone tissue proper. The bone tissue, first part of the bone tissue proper facing the periosteum is the compact bone. Then the part of the bone tissue proper facing the endosteum is the spongy bone. So this is the spongy bone here. And then we have the endosteum ending that. So compact bone or cortical bone is has an elaborate architecture where the lamina is really well organized. They are organized into three different manner. They are organized circumferentially. They are organized into what we call an avation system, and they are also organized into an interstitial lamina. I'll show you a picture of how that looks like. But it's important to note that the compact bone, unlike the spongy bone, doesn't have spaces between them. They contain large amount of these bony tissues that are organized in layers, lamellar layers, and into avation canal, and makes it a very, very compact and tight as a result. So this is just a picture showing us the compact bone. So compact bone, as I said, have very organized layers. So you can see, first of all, the first layers are these are the laminar layers or organized or of an organization for the compact bone. And if you also notice, you see these circular structures. This is the avation canal. So circular structures you are seeing here is the avation canal. So this entire portion shows the compact bone. This entire aspect that I'm describing here, it is the compact bone. The compact bone makes the diaphysis of the long bone. It contains continuous sheets of uninterrupted bone, and it helps in protection and support of the bone. It resists stress being applied to the bone. So the compact bone is the strong part of the bone. And you notice just after the compact bone, you see this um, network of bones. This is the spongy bone. I'll talk about that in the next slide. So the spongy bone, as shown before, is the part that makes the epiphysis of the long bone. It's also found in some of those other flat bones I mentioned, like the skull and the sternum. It consists of a lamina that is built as a form of network, what we call a trabeculus. You can see the spongy bone being formed in networks. You can see the trabeculae. These are network just connected together. And you can see the spaces between them. All the spaces there is what makes it to be known as the cancellous bone or spongy bone. The last layer of the bone is known as the inner layer on the endosteum. It is composed of the progenitor cells, the osteoblast, the osteoclast, and the bone lining cells, as mentioned. These cells tend to line the free surface of the spongy bone. They line the trabecula, and they line the avation canal of the compact bone. And as a result, they are found in the innermost aspect of the bone. Let's end the entire part of the aspect of this particular model under bones by talking about the formation of the bones. The process by which bone is formed is known as ossification, and we have two major ways in which bones can undergo ossification. We have what we call intramembranous ossification, and we have what we call endochondral ossification. Starting with intramembranous ossification, this involves the direct replacement of a mesenchyme to bone. So most of the connective tissues we have is gotten from mesenchyme. For us to form bones, we have to form them from mesenchyme. So when we have a mesenchyme forming a bone directly, we term this as an intramembranous ossification. And how does this go about? So usually the mesenchyme cells would aggregate first of all. Once they aggregate, blood vessels will appear within the aggregates. These blood vessels will give nutrients and important factors, growth factors to the mesenchyme and allow it to be to undergo ossification to form bones. What we start to see is that we start to see osteogenicity being invoked in this aggregate of mesenchymal cells, and the mesenchymal cells will become or start slowly become osteoblast as a result of osteogenicity. So once the mesenchymal cells aggregate and blood vessels come into their mix, the cells are converted into osteoblasts. Once osteoblasts are formed, the osteoblasts will start to now secrete that extracellular matrix that I talked about, the immature extracellular matrix, which we know as osteoid. So the osteoid is formed from the osteoblast, and then slowly the osteoids now become mineralized. So this involves the accretion of the organic and inorganic compound of the ground substance into it. So once the osteoid becomes mineralized, remember that the osteoblast becomes osteocyte. Then we now start to see the matrix being formed. So the matrix being laid down and organized. So the organization of the matrix starts to occur. So this will be laid down in a initially as a woven form and then eventually as a laminar form. So laminar matrix are formed and then they will fuse together to form different aspects and fuse together to form a regularly arranged lamella in the compact bone or an aviation canal in the compact bone. Or they can be arranged or fused together to form the trabeculae of the spongy bone. So trabeculae and the spongy bone is formed, is interweaved, wove together, and as a result, they give rise to the spongy bone. For the compact bone, the matrix are arranged elaborately to form the compact bone architecture. So this is practically how intramedullary ossification would follow. We have mesenchymal cells aggregating together, blood vessels entering into them to supply nutrients, and then triggering osteogenicity. Osteogenicity will lead to the conversion of the mesenchymal cells into an osteoblast. The osteoblast form will start to secrete matrix, immature matrix called osteoid, and then from there they will mineralize the matrix to form a mature matrix. Once the matrix have been mineralized, the matrix will now further be arranged or organized into either a lamina, into lamellas, and the lamellas could either be a trabeculae or a regularly arranged lamella, so to form either the compact bone or the spongy bone. So this is just the image of how the intramembranous ossification tends to occur. You can see that we start from mesenchymal cells that become osteoblasts. Osteoblasts will start to undergo mineralization to form osteocyte and the osteoid, and then from there this slowly becomes the lamella that we talked about. You can see it becoming compact bone and then trabeculae in the spongy bone. The second process of formation of bone is known as endochondral ossification. And this one is very distinct from the intramembranous ossification because we go from mesenchyme to cartilage to bone. We don't go directly from mesenchyme to cartilage, mesenchyme to bone. And by virtue of the name, we call it endochondral because we are going from cartilage to bone, and that's the key thing to remember. So this starts first of all with the formation of a cartilage. And once the cartilage is formed, how the cartilage undergoes endochondral ossification is by multiple processes. So it usually starts first of all by the cartilage undergoing a loss of its supply of nutrients. So the nutrients are tend to diffuse into the cartilage is interrupted. And once we have that occurring, two things would happen to the cartilage. The cells of the cartilage will start to undergo hypertrophy in response to the loss of nutrients. And with time, once they undergo hypertrophy that is not sustainable, the cells start to die. So the cartilage starts to undergo degeneration. So two things happens to the cartilage once nutrients is being cut off. It undergoes hypertrophy, and then with time, it starts to degenerate. As it's degenerating, the cells they are being lost, and it starts to produce a space. As this is occurring, it's important to note that the extracellular matrix that is surrounding the cells of the cartilage undergoes calcification. They are calcified. All right. So as this is occurring, as this cartilage is degenerating and extracellular matrix is calcified, we start to see a space appearing in the cartilage because it's degenerating. So a space starts to appear, and that space is known as the primary marrow cavity. All right. As the space appears, we start to now see blood vessels entering into the space. So the blood vessels start to appear within the disintegrating cartilage and enters into the primary marrow cavity from there. So slowly, as the space, the primary marrow cavity is formed, from there we see, and then the blood vessels start to enter, start to migrate into this primary marrow cavity. Slowly, we start to see mesenchymal cells entering into the cavity. These mesenchymal cells will become osteogenic or osteogenic cells or osteoblasts within that cavity. And then slowly, the osteoblasts that have migrated into the cavity starts to deposit osteoid. So they are depositing the matrix, and as they deposit the matrix into the degenerating cartilage, into that primary marrow space, the matrix that is deposited is usually immature. It becomes what we call the osteoid. Osteoid there becomes mineralized, and as the osteoid becomes mineralized, it will give rise to a primary endochondral bone. So let's recap it again. We start with cartilage losing its nutrients, becoming hypertrophied, and then automatically becoming degenerated. So it loses its substance. Then we have that degeneration leading to a space being formed. We have a space being formed called primary marrow cavity. That space is now filled with mesenchymal cells, and these mesenchymal cells that we are seeing here will start to undergo changes to form an osteoblast. Once the osteoblast is formed, the osteoblast within that primary marrow cavity starts to produce matrix. They produce an immature matrix that is called osteoid, and that immature matrix becomes mineralized. Once the immature matrix of the osteoid becomes mineralized within the primary marrow cavity, we call it that the primary endochondral bone. So primary endochondral bone is mineralized osteoid found within the primary marrow cavity. So the primary endochondral bone is eventually now reabsorbed by osteoclast and chondroclast. And once it is reabsorbed again, this gives rise to another cavity called secondary marrow cavity. So we have two cavities being formed. The primary marrow cavity formed when the cartilage degeneration, and then the secondary marrow cavity formed when the initial osteoid that was deposited becomes reabsorbed. So this secondary marrow cavity, once it has been formed, we now have another set of um, matrix being laid down. So we have matrix laid down twice. So the second or new matrix made laid down is now known as the secondary endochondral bone. And at the end of the day, we have that bone being formed. So you might wonder why this entire process. Um, why are we having two different points where we have bone being laid down? This is just to ensure that the entire cartilage architecture is converted to mature bone at the end of the day. Remember that this is not similar to intramembranous ossification, which we just go from mesenchyme to bone, and then the bones are reorganized to form lamella. This one we are going from cartilage to bone, so we're ensuring that whatever bone is laid down is strong enough to be an actual bone at the end of the day. So we see this process occurring in a number of bones, especially long bones, and usually they tend to undergo different forms of ossification. And so we have them undergoing primary and secondary ossification at different points. So this is a picture that depicts for us the endochondral ossification. You can see how we go from a single cartilage that degenerates at the center to form that primary marrow cavity, and then primary marrow cavity has blood entering into it, and then this will mineralize to form bone as shown here. And then from there, we have that bone also forming another cavity there, secondary marrow cavity, as shown here. And that bone, with time, would also mineralize to form secondary marrow bone or secondary precordial bone. The last aspect of this model is on cartilage. Cartilages are specialized connective tissues with high tensile strength and elasticity, and they're found widely in the entire body. So cartilages are found in areas like the ears, the nose, the movable joints. They tend to line the articular surfaces of the joint. Remember, I said that the articular aspect of a bone is not lined by periosteum, rather it's lined by cartilage. They are also found in bronchial tubes. They are found in rib cages, a intervertebral disc, and in the pubic symphysis. Cartilages are divided into three types. We have the hyaline cartilage, we have the elastic cartilage, and the fibrocartilage. We'll talk about each of them. So this is a picture of each of the different cartilages we have. What tends to distinguish the cartilages tend to be the components of their extracellular matrix. Remember that cartilage would also contain cells, contain extracellular matrix that is divided into ground substance and extracellular fibers. So the cells within a cartilage include things like chondrocytes and chondroblasts. And the extracellular matrix within it would contain ground substance and most especially fibers. Fibers would either be collagen, elastin, or reticular fibers, and this tends to distinguish the cartilages into hyaline cartilage, the fibrocartilage, and elastic cartilage. And how they arrange also tend to determine. So hyaline cartilage tend to have a clear looking ground substance. That's what makes it hyaline cartilage. Clear looking ground substance. So it has very little extracellular fibers, but large amount of ground substance that is clear. The fibrocartilage has densely packed collagen fibers within it. So the collagen fibers are larger in the fibrocartilage compared to the hyaline cartilage. So they have large amount of collagen fibers and reticular fibers, and they are well arranged and well organized into different rows. So the cells themselves are also well arranged. So the cells in the hyaline cartilage are sparsely distributed. Cells in the fibrocartilage are well arranged. They are arranged into flattened rows of cells. Then we have the elastic fiber and cartilage. Elastic cartilage is one that contains large amount of elastic fibers. So elastin is found as the extracellular fibers compared to collagen in the fibrocartilage. But these elastic fibers are woolly arranged. You can see how they are irregularly arranged within the elastic fibers, and this allows the elastic cartilage to be able to undergo distortion and reformation of its shape. Where are the sites in which we could have the different forms of cartilage? So, hyaline cartilage, for example, is found in areas like the joint. See hyaline cartilage on the joint. It lines the articular surface of most of the joints, the knee joint. You can see how this whitish area of the knee joint is lined by hyaline cartilage. Elastic cartilage is found in areas like the ears. If you flip your ear, you see that it is very elastic. It reforms back when you distort its shape. And then fibrocartilages are found in areas like the intervertebral disc, where there's a lot of stability and strength needed or necessary. And remember that fibrocartilage contains large amount of collagen that maintains this. And that concludes our module on connective tissue 2.